Well, it's lovely to be here, and I'd like to thank the organizers for thinking of me and of the CGIAR and what the Gates Foundation is trying to do to help deliver better cultivars to breeders and to farmers primarily we focus on sub-Saharan Africa and South Asia, and that's where I'll be focusing my attention. The advent of low-cost, mid-density genotyping opens up many new opportunities for applying the genomic relationship matrix to accelerate genetic gains in plant breeding. But pipeline design in plant breeding programs really hasn't exploited most of these opportunities yet. I've been working closely with Christian Werner and the other CGIAR quantitative geneticists, a number of people in the room, to assess these opportunities, in particular sparse testing. And I'll try and run through them with you today. And how do we advance the slides? No, that's not advanced. There we go. Yeah, and you'll be able to see very quickly that my slides are completely uncontaminated by suggestions from ChatGPT or any other formatting support. I'm going to talk about three main areas: design flaws of conventional plant breeding pipelines in terms of maximizing genetic gain. Plant breeding programs are more designed to extract products quickly from a particular segregating population than to increase performance of a population over time. I'll run through three use cases showing how GRM-enabled sparse testing can help overcome these flaws. And I'll describe a pilot experiment using sparse testing methods to conduct genomic selection model training first stage agronomic evaluation on farm in a SIAT bean breeding program in Tanzania. Plant breeding, so in terms of how plant breeding programs are designed, they have two basic components. They've got a cyclic population component where, recombinant progenies are generated, phenotyped, and then the best selected and recombined to form the next generation. And then there's from every cycle usually, the best selections are advanced into an evaluation pipeline for potential commercial release. And there are really very different objectives for these two phases. The population improvement phase selects for breeding value, or it should select for breeding value. Parents for each cycle need high breeding value, but they don't need to meet all the minimum trait thresholds of a target product profile for commercial release. On the other hand, commercial cultivars need to meet all those minimum thresholds, but they don't need to have the very best breeding value. And only the population improvement component of this system for additive breeding value contributes to genetic gain over cycles. The system is often represented as a funnel where you've got, recombinant progenies are evaluated through a series of steps, usually with increasing accuracy, more locations of testing, and then finally into a release process. And at some point in that funnel, which is represented as a kind of a gentle fluting, some selection candidates are moved back into the nursery for the next cycle. But really, that funnel should look more like this. Conventional plant breeding programs often will evaluate, the programs that I work with in the CGIAR will often evaluate something on the order of 10,000 selection candidates in a nursery at very low accuracy. So I'm trying to represent the repeatability here at a single location. Then they'll move, winnowed down very quickly to a few hundred selection candidates and evaluate those at in stages, cutting about half each step with increasing levels of replication within and across locations. But about 99% of the total selection pressure is applied at 1 to 2 locations and very low accuracy. The GRM allows us to reconceptualize that process, replacing information from replication of selection candidates with information from relatives using a sparse testing approach, which was, I think the first simulation and description of sparse testing was in Jeff Endelman's paper in 2014. But I think the term comes from a conversation that I had with Jean-Luc Yannick in his office a few years before that. Sparse testing treats the haplotype rather than the clone or the line as the selection unit. Contrasting haplotypes can be dispersed across different background genotypes in segregating families. Sparse designs are created by dispersing related candidates to different locations, farms, or onto different hybrid parents in a hybrid program. And I'll be talking about the first two applications, dispersal across the sample of the TPE. But I would refer you to a poster by David Gonzalez to look at options for using the same approach in hybrid breeding. Allowing relatives to substitute for reps in sparse designs permits a really basic redesign of a population improvement component of plant breeding pipelines, allowing us to really focus on selection for breeding value. I'm going to consider 3 use cases for sparse testing. The first would be shortening the breeding cycle by eliminating the need for seed increase steps. If we don't need to replicate a selection candidate over many locations and replicates, we don't need often a seed increase step, which actually sucks up a year of the breeding cycle in many cases. It also allows us, by permitting or achieving higher accuracy in the early stages of selection, it allows us to select parents reasonably accurately from in fewer years of testing. For that to work, we really have to be able to achieve a pretty good level of accuracy in early generation selection. So that's achieved through better sampling of the target population of environments at early stages when, as we've seen, the great bulk of selection pressure is applied in plant breeding populations, normally at low accuracy. And then finally, I'd like to consider a use case where we're dealing with systems often, particularly in sub-Saharan Africa, where conditions on the research station are really different from conditions on farms. Farmers are very resource constrained and managed differently than the environments in which their varieties are selected. And as a consequence, we don't know much about this, but it's possible that the genetic correlation between performance on the research station and effects estimated on farm is low. So we can use sparse testing on farm to deal with that problem. So I'll use these blob diagrams here to describe the use case of increasing early stage TPE sampling. So the big blue blob is meant to represent the target population of environments. The little green blob is meant to represent a research site that's sampling those environments. And in a typical program, a nursery might evaluate something on the order of 10,000 candidates without replication and probably without, for example, yield measurement at a single location. It'll be visually selected for adaptation. And then that population will be severely cut. An initial agronomic trial in the programs I've been working with, often at that initial agronomic replicated trial will be at 1 or 2 locations, often 1. And maybe so having invested about 10,000 plots in the initial step, these programs will invest something on the order of 1,000 plots or less in the second step. And then go through several stages of testing and selection. A conventional third stage might involve testing 50 remaining selection candidates at something like 20 locations, often with some replication within the location. And so these advanced steps will focus an awful lot of testing resources on very few selection candidates. That's pretty expensive to establish those networks, those multi-location testing networks in time and organizational capacity. But they're absolutely critical and underutilized resource in many plant breeding programs. So, and they present an opportunity for us to really change how we think about that stage 1 trial. As instead of a winnowing step, a selection step, we should be viewing it now as a genomic selection model training step. And we can do that using sparse testing by simply taking our stage 1 selection candidates and adding them to those locations where we're already doing multi-location testing. There's a cost to this, but most of the costs of establishing a remote location or working with a partner for them to manage the location are the costs of setting up the location and managing it. Adding a few plots per location is usually not a huge cost. And this blob diagram is meant to represent that situation where we've added 40 selection candidates maybe to each of our 20 locations, allowing us to take a set of related lines from a population and get them phenotyped and get that data into our model. This has been simulated in pretty comprehensive terms by Christian Werner and some of our colleagues in the CG Quantitative Genetics Group, which is really pioneering the description of these designs. And I'll present and hopefully not butcher the results of Christian's simulation work. But to get the real story, please have a look at the preprint on Biometrics. And we settled on a series of simulations that tried to use equal testing resources. That's a little bit complicated to do, but each of the scenarios that Christian looked at were involved allocating 220 plots, 2,200 plots to different combinations of selection candidates, locations, and replicates. So we looked at, he looked at 3 population sizes, 220, 440, or 550 candidates from a total of 22 crosses with different numbers of individuals per cross to make up the total number of selection candidates. Different, in the conventional testing schemes, 2 to 5 locations, replicated or unreplicated, number of plots per site ranging from 440 to 1,100. And so these 3 scenarios were run without generating a GEBV from the genomic relationship matrix, with the GEBVs, and then a set of complementary sparse testing scenarios that involve 2,000 genotypes partially replicated at 10%, allocated or divided among either 5, 50, or 440 locations. So with that partial replication, and compared with the treatment that called the control, which was these 2,000 genotypes evaluated as a single location. And each scenario involved 2,200 plots. We'll just focus on the right hand, my left side of this equation, of this figure. Gain that was achieved on average across simulation runs over 10 cycles of selection. I think this is expressed in genetic standard deviation units, right? The conventional scenarios are these three lower scenarios. Adding the genomic relationship matrix into those, selecting on the basis of GEBV, increased gains modestly, but with no cost except for the genotyping. It's worth doing that. But the sparse testing designs increased gain by about 20% in total over the program. And interestingly, that most of that gain was achieved with the scenario where we distributed the training population across only 5 locations, but there was not there wasn't a big benefit or cost in terms of genetic gain to distributing them across 400 and something like 400 locations. And why we're interested in so many locations for that particular scenario, we'll deal with in a second. The second major use case is shortening the breeding cycle. That's very straightforward, especially once you've got a way to get accurate estimates of breeding value from stage 1. Generally speaking, the nursery stage, you might, a conventional program might take 1 year to evaluate, 1 year to do seed increase, and then move material through increasingly accurate field testing. I should have put the point of the arrow up here so that we're not, we're recycling back into the nursery, of course. If you've got accurate ability to select for breeding value at stage 1, well, you can, first of all, cut out that season of seed increase, which particularly in clonally propagated crops is almost unavoidable. And you can select your parents and get them back in the nursery after stage 1 evaluation, updating your prediction model. So sparse testing allows accurate selection of parents from stage 1, and this has the potential to reduce the breeding cycle by 2 or 3 years. The final use case that I'll describe is the moving model training on farm in situations where you've got a very nicely managed, probably irrigated, fully fertilized research station environment in which most selection is occurring, but the products are being used by farmers who are investing much less in fertilizer and who are often using much lower plant densities, have lower or less effective weed control. And so the question really arises, what is the, how well can we predict performance in this environment from testing and selection in this environment? And it's a surprisingly under-researched question in plant breeding for in situations where farmers are really constrained for inputs. So those differences in management on station and on farm may lead to lower correlation, and the genomic relationship matrix and sparse testing approaches allow us to generate breeding values in farmers' environments directly. And so you know, the idea then is to take your stage one, and instead of testing them all on the research station, moving them immediately into on-farm trials. And we'll discuss a little bit how that can be done. Under what conditions will testing on-farm produce greater genetic gain than testing on the station? Well, we can model that using the classic approach to describing the response to direct versus indirect selection, where the ratio of direct response response to direct selection on farm as a proportion of the correlated response we would expect on farm to selection on the research station is a function of accuracy of selection on farm versus accuracy of selection on the research station, and the genetic correlation between effects estimated on farm and on the station. So yield under farmer management is our target trait. In the UK and in Canada and the US, management on farms is usually as good or better than on the research station, so we don't worry very much about that genetic correlation. In sub-Saharan Africa, I think it's worth worrying about. So how well the station predicts breeding value on farm is a function of that genetic correlation and the relative heritability on station versus on farm. If accuracy is way better on station than on farm, then even with a relatively low correlation, you would want to select on the research station. There might be other logistical reasons. But if reasonable accuracy can be achieved on farm and that genetic correlation is low, you would expect a substantial advantage to breeding values that are estimated on the farm. Yeah, I've already described that. Now, what are the practicalities of getting breeding values estimated on the farm? You have to treat individual farms as incomplete block, so you take your training population and you spray it out across a large number of farms, we're often dealing with very small farms in very heterogeneous environments, farmers with very different capacity to access production resources. So careful sampling of farm and farmer heterogeneity is is needed to make sure that your model is accurate. And when we first started doing this, for reasons of simplicity and logistical ease, we would often try to allocate something on the order of 20 plots, sometimes, and for late stage on-farm testing, that's still often done. People try to replicate on individual farms, for example, a relatively small number of selection candidates. But we found that this, trying to find farmers who can handle 10 or 20 plots means that you're excluding the poorest and most disadvantaged farmers. And so we've really moved to what I would refer to as farmers incomplete block designs, where only 3 to 5 plots are allocated to an individual farm. And that's been fairly recently branded as the tricot approach, but it's been floating around for a long time. Using farms as incomplete blocks, going back to the blob diagram, allows us to move from sparse testing on the research station, where we take our training population and spray it out across perhaps 20 test environments, and we move those, we evaluate those selection candidates on individual farms, each, several hundred farms, each with somewhere between 3 and 5 entries. It's fair to ask, how do you, what are the logistics of that? How can that be managed? Well, I'll talk about that if we have time briefly at the very end. I think we're still okay for time. But let's move on to a pilot. We're piloting this type of approach in 2 breeding programs. One is the Siat yellow bean pipeline in Tanzania, led by Teshale Memo, based in Arusha. We're also working on it with the Simit maize program in Eastern and Southern Africa. I'll present the bean, the results of the first year of bean work today. We've got 2 years of results from maize, but it's a more complicated story with several heterotic groups, and we had essentially a failure in the second year with as a result of the El Nino drought in 2024. The bean experiment consists of 419 breeding lines and 36 release varieties that have been genotyped with about 1,200 SNPs, and they were allocated to, I think, 4 plots per farm on 309 farms. There's an equivalent, there are 3 main bean growing regions in Tanzania: the Lake Zone, the Southern Highlands, and the Northern Highlands. Teshale organized testing at 103 farms in each of those zones with more or less equal distribution of lines across those trials. There are also 3 research stations on which the population was evaluated so that we could compare heritability and correlations. 2 of those locations are in the Northern Highlands. 2 are in the Southern Highlands, and there's not a station in this network in the Lake Zone. The design was partially replicated on the research stations. Some of the parameters from those trials, estimated from those trials, are presented here. So first of all, you can see that there was a big mean difference between the on-station and on-farm trials. As expected, yields were a lot higher on the research station, nearly twice as high. Interestingly, heritability accuracy was a little bit lower using the for the GBVs on the research station, and great appreciation to David Gonzalez for getting this analysis done and presented clearly. We've estimated the genetic correlation between the effects estimated on station versus on farm as about 0.44. So relatively low, but thankfully positive correlation between performance on farm and on the research station. Plugging those estimates into the classical model, we would expect gains from direct selection on farm to be about twice what they are from selection on the research station. Of course, we're not advocating even, and this is preliminary results, a single year of testing. We're repeating these experiments over several years, but these will be important parameters for us to have a better understanding of. We're not advocating abandoning testing on the research station, even at stage 1. There are many types of screen that are best done on the research station for disease resistance, quality, other types of traits. But Moving this testing stage on farm also has many important advantages. It allows us to get input from farmers on their perception of the value of the candidate early on and to model that. So, we think that this has potential for incorporation into standard plant breeding practice. How difficult and expensive is it to do this type of training on farm? The cost is, it's a serious undertaking, but it's something that programs need, and there's a pretty steep learning curve, but it's manageable if the trials are not implemented by CGIAR breeders. They have to be implemented by organizations that are working in villages on agricultural development in some way. Organizations that are used to getting out to a lot of farms and interacting with a lot of farmers. Typically, these programs that are intensifying on-farm testing hire local high school or university graduates from the villages or the communities surrounding. Both CIMMYT and CIAT have been pretty successful in getting high rates of data return from these trials at an affordable price. I'll wrap up with these conclusions that sparse testing, supported by the GRM, permits extensive sampling of the target population of environments during stage 1. The stage in breeding pipelines when GS models should be trained, and the stage at which typically most selection pressure is applied. You'll notice actually that sounds a lot like dairy cattle breeding, so that's we're learning a lot from the animal breeders. This increases accuracy and reduces the effects of variable trials. I didn't discuss that very much, but when you're basing most of your phenotypic your phenotyping activity on one or a few locations, you're really at the mercy of the weather at those locations. If you are able to spray your training population out across your target population of environments in 10, 15, 20 locations, we would expect that you'd get a lot less jerking around of estimates of breeding value from year to year. Sparse testing at stage 1 doesn't need to increase costs if candidates are dispersed among all the locations that you're already using for variety testing. It does increase transaction costs a little bit, but there are I suggest that you chat with Clay Sneller here about trying to implement this type of network in a series of or a group of independent wheat breeding programs in the eastern U.S. It allows the breeding cycle to be shortened by reducing the need for seed increase for replication of selection candidates and by allowing parents with high breeding value to be selected after stage 1. And it permits, if we apply farmers' incomplete block designs, we can get an estimate of breeding value for on-farm performance from stage 1 and apply that in genomic prediction. We see that, generally speaking, the accuracy estimated from these farmers' incomplete block designs is a little lower than you get on good research station trials, but it's not massively lower. And the approach has special applicability to situations where you really, you've got very heterogeneous farmer populations, and those farmers really are managing under different conditions than you experience on the research station. shorter breeding cycles, better sampling of the target population of environments, and more accurate representation of the conditions under which farmers are working have the potential to substantially increase genetic gain. So that's an overview of some of the changes that we're trying to support in the Gates Foundation investments in plant breeding. We're not only focused on improving population improvement. It's extremely important to be delivering products where breeding programs in the CG and elsewhere are under tremendous pressure to deliver those products. But we're not going to get them unless we improve the source populations. Thanks.